today we have Dr Ralitsa Madsen. So I was lucky enough to meet Ralitsa during her early career when she worked with Rob Semple in Cambridge, where her and a team in the lab discovered my PI3K mutation, which I'll always be very grateful for, so thank you.
I wasn't part of that, Mandy, though. I'm not taking credit for that.
No, you can take
Vicky.
it out later. But since then, Ralitsa's gone on to dedicate her research into PI3K, both within cancer and PROS. Her research, it aims to understand more about the PI3K gene and how it affects the cells, and to use this knowledge to identify novel therapeutic options for those living with PI3K mutation. We're also extremely grateful to Ralitsa for her continued support for the charity and her contribution to our annual newsletter with yearly updates, yearly updates into her research into PI3K and PROS, which is a valuable part of our newsletter, you know. So please welcome Dr Ralitsa Madsen as she gives us some research updates into PI3K.
Thanks, Mandy. Hi, everyone. Really happy to meet you all. And I think I've met you sort of virtually in on various occasions, if nothing else than Twitter for Mandy. It's really hot in here, so if I get really sweaty, it's not because I'm nervous, but it's, if anyone else is hot, maybe it's not just me. Right, so I think, Mandy, you've put my title to be PROS Research Update, and I actually changed it slightly to PI3K Research Update. It's very difficult for me as a scientist to say PI3K because I always want to use the whole scientific jargon, so PI3 kinase and even adding an alpha at the end. So if you hear me say some of these extra scientific words, just remember it's all PI3K. So I'm a researcher, I'm not a clinician, and my passion really lies in understanding how things work, how PI3 kinase works, and how it drives disease. But really, at the very mechanistic level, so nitty-gritty details which are important in order to understand how drugs work, why certain people get one symptom, others get another, why the drugs work in one patient but not in another, and things like that. That all needs to come from basic research understanding of how this circuit in our cells actually works. Fortunately, I also recently managed to establish myself as an independent group leader, have my own research lab now. So I've just moved up to University of Nudity, more specifically this particular center called, very annoying long name, MRC Protein Phosphorylation and Ubiquitination Unit, just MRC PPU. But the reason I mention it is because this is the place where a lot of the PI3 kinase sort of components, you know, when you look at PI3K, you've heard about AKT, you've heard about mTOR, the mechanisms of how those bits work, a lot of those mechanisms were actually discovered and mapped up in this place. So for me, it's the perfect place to be, and I have a lot of new colleagues who are equally passionate about, you know, driving basic disease understanding. So hopefully we'll be able to make some progress in that space. In terms of the talk today, I'll give you 3 sort of research updates, more generally, not just related to my own work. And then I'm going to switch gears slightly to some new bits that I have discovered over the last few years, things that are not yet in the public domain, but hopefully will be in the public domain within the next 6 months. I've been saying this for the last year, so I'm hoping actually it will be before the end of the year anyway. The first update, I already gave it to you, but obviously for me, big news from the past year is that I've started my own group, and it's been quite a journey, but this is a picture of me as a first-year PhD student in Rob's lab, September 2016, and this is Rachel Knox. I think some of you may have come across Rachel. She was an excellent research assistant in Rob's group and was driving a lot of the genotyping or sequencing of patient samples, and has also been involved in some of the newsletter writing back in the day. So this is the first time we sort of got really exciting PI3K data from my own research as a PhD student in Rob's lab. And I should say for the last 7 years, we continue to have exciting data, at least in our view, and now it's not so much me as a student, but I now got a really dedicated student. Actually, both of them are great, but Gray has left because she's moved to new things. Olivia has joined my lab. Some of you may have come across bits that Olivia has written. I'm going to mention some of that later on. And these are messages that Olivia was sending to me at 2 AM 2 days ago with some really exciting data. Not because I asked her to work at 2 AM, but she was so excited about doing the science, and I'm really lucky to be working with somebody like that in the lab. So the excitement basically continues. And other news that I think are relevant to this community is the fact that we as researchers are starting to establish a community of our own and raising awareness about PROS within the research community. So as I said, I call it PI3K research because PI3K is involved in many diseases, okay? So normal body function relies on a functional PI3K. And therefore, there are many people in the world who work on something related to PI3K and even PIK3CA. It's very common in cancer. And that knowledge some of that knowledge could be transferred to improve our understanding also of PROS. So it's really valuable that the CLOVE syndrome community, which is led by Kristen Davis in the US, has now enabled this initiative where we're meeting once every 2 months as part of a PIK3CA related conditions research roundtable to discuss research. So it's really focused on research. It's not very lay people friendly in the sense that if you join you would be bored to death, but it's really good for researchers to be able to share and to collaborate with each other. And the key thing behind this research roundtable is that we want to share results before they're in the public domain, because you may or may not be aware, but scientists can be really annoying and secretive about their findings and can take forever to get stuff out, and that's not to your benefit. So if we have a forum where we can share results earlier and make progress that way, that's better for us, but it's also better for you. So that's an initiative that's ongoing. The third thing, of course, is the fact that more and more opportunities are emerging for treating or repurposing drugs that have a beneficial effect in the context of PI3K disorders. And of course, a lot of that has been driven by the fact that this is one of the most commonly abnormally regulated signaling circuitries in cancer, okay? So a lot of research on drugs to target PI3K and some of those other components come from cancer. And we've had, thanks to that, we've seen that the FDA, so the Food and Drug Administration in the US, approved alpelisib for PIK3CA related overgrowth spectrum. And that followed from the fact that alpelisib was approved for treatment of estrogen receptor positive breast cancers in combination with another drug. But again, advance in another research field focused on PI3K benefits advances in PROS. Rob mentioned sirolimus, and he did already provide us with quite a bit of detail in terms of the benefits and some of the side effects. Now, sirolimus acts on this thing called mTOR, which very simplistically, and I always cringe when I see these pictures, but I know I have to do it to keep it simple, but very simplistically, mTOR is somewhere further downstream of PI3K. So you have active PI3K, and if you have that, you may also have more aberrant activity in mTOR, and that's why sirolimus can be beneficial, okay? And it turns out it can be particularly beneficial in slow flow, what's so-called slow flow vascular malformations. This is something that was out last week. Again, as Rob said, it's really important to be critical of these studies that are not registered, it's registered but are not randomized trials, okay? And then something that is not related to PROS yet, but just 2 days ago, the Food and Drug Administration approved capivasertib in combination with another compound for treatment of breast cancer. So again, this is similar to what happened with alpelisib before it made it into, it was repurposed for treatment in PROS. So capivasertib is a drug that hits AKT, okay? So again, PI3K, AKT, mTOR, this is what undergraduate textbooks typically show, even though it's it's kind of right, but it's also partly wrong, but I'll come back to that. Anyway, AKT is the one that can go wrong in PIK3CA syndrome, which I think some of you may have thought you have had, and so you've got PIK3CA sequence. So it can also be beneficial, potentially, in the context of abnormalities in this circuitry. So why am I a little bit hesitant? Well, the reason is that, this is good, it helps, it can help. Some of you have been trying alpelisib, sirolimus, but you also know that many of you don't actually benefit, or maybe you have too many side effects. So we still need more treatments, or we still need better, or maybe better ways of using the treatments that we have. So that's why there's still more to be done. And one of the reasons why there's still more to be done is also the fact that at the moment, the way we're using these drugs here is really very much like a hammer. So those are blunt tools. You are switching things off. You are not making them work. You are switching them off. So you're removing bad things, but you're also preventing them from doing the good things that they're supposed to do in your body, okay? So although it's very convenient to think about PI3K as a switch, it is not correct from the scientist's point of view. If you're sitting and working with PI3K every single day, this is not a switch. It is not as simple as just, okay, PI3K is on, growth, PI3K is off, no growth. You know, that's kind of typically how it's presented, but there is much more complexity to it. In fact, it's a very complex beast, and the way I tend to represent it is the Ralitsa accordion. So this is my view of how PI3 kinase, I call it signaling. When I say signaling, this basically reflects this relay that happens in the cell when you have PI3 kinase activity, then stuff happens. Imagine lots of wires. Instead of wires, we have biochemical small molecules. So PI3 kinase signaling to me is a lot about context. Whatever happens when you activate PI3K here depends very much on which cell are we looking at, what developmental stage, what is the environment. I mean, that part also depends on which cell you're looking at, because different cells will be part of different tissues, different environments, different signals, and also which mutation one has. So for example, Rob mentioned some of that already. So to me, the way I think about this is as a very complex communication device, and the output of this communication device is context dependent, okay? So if we want to understand PI3 kinase signaling, if we want to target this better when it goes wrong, we need to understand what kind of information does the PI3K carry. So think about it as a communication channel, right? So there's an input, there's an output. How is the input translated into an output by PI3K, okay? And can we tweak that without switching it off, okay? So that's the aim of my lab, is to solve what I call the PI3K communication code. And it is no coincidence that I'm showing you a picture from the principles of human telecommunication, because we're actually applying some of that theory to understand or analyze some of the data that we are getting in the lab. And really what we're trying to do is listen in on the waves, you know, the signals, and trying to understand what different signals mean in terms of the output or the function that a cell then executes. So I'll show you in a second how we can do that. But essentially, your PI3 kinase sits here. It's the channel, it transmits. So if it doesn't translate the signal correctly, well, then you have a problem. So imagine on a noisy telephone line, someone is calling you and you don't understand, causes communication problem. Maybe someone was supposed to say yes, but you heard no, and then you do something that you were not supposed to do. You know what I mean? So how do we do this? Well, actually, I should explain this with another much simpler analogy, just to give you a sense of why the telecommunication analogy is so useful. So if you think about a cell, our cells receive signals all the time. So you've got, the cell is bounded by a cell membrane, and within the cell membrane you have these things like little antennas, and they receive all these different signals. You have different antennas for different signals. The key thing is that your PI3 kinase is used by pretty much every single one of these antennas. It's there and it's listening in on what's coming from these antennas, and then it determines how each one of these signals is interpreted further downstream by all these other workers like AKT, mTOR, for example. And because it's such a central, what we call signal transducer, if it goes wrong, then a lot of things further downstream can go wrong. And that's your mutant PI3K. That's where it comes in. And so I mentioned it already, we need to understand what this code is, because rather than switching the system off altogether, maybe we'll be able to tune it back to normal instead. So what I've spent a lot of time doing over the last few years is developing new methods to record the waves, the PI3K signal waves, and really try and map, if I have this signal, what is the pattern? If I have this signal, what is the pattern? If I now have a mutation, what is the pattern and what does it mean in terms of output, and how can I correct it back to normal? I can't do that by, you know, recording phone conversations between cells, because it's not how it works. But I spend a lot of time in darkness staring at cells that are colored, and basically we can actually measure activity of PI3K waves by looking at how we have a particular, what we call a reporter. So it moves in and out of the cell nucleus or the cell control center, depending on the activity of PI3K. We can quantify that, and then we can start to, do clever sort of modeling further downstream and understand what is the code that I talked to you about earlier. So next slide is going to be heavy on science. So I'll ask you to only follow what I point at and nothing else. So ignore IGF-1, EGF, EPI. What that means is basically signal 1, signal 2, signal 3. So this is me challenging, having cells in a plastic dish, and I'm challenging them with different signals that I know they may be seeing in the body, okay? And I have cells that are either normal or cells that have a PI3K variant that many of you may have. It's the H1047R variant. Now, when I challenge them with these different signals, I then record the waves, okay? If you just look at the black traces, or even the gray traces, they're overlapping. These are the cells that don't have the PI3K variant, so they have the normal PI3K. And you can see that for each of one of these different signals, you have a distinct wave. So you have a distinct pattern. It's a bit like different letters or words mean different things, right? So this is that pattern, this signal is this pattern, this signal is this pattern. Now, in orange and red are the cells that have the PI3K mutation. And what you will notice, if you look carefully, or have a lot of time to stare at this like I do, is that these patterns are now much more similar across these three different signals. So they're overlapping, they're not distinct, they're like blurred. Can you see it? Or am I the only one who can see it? So I'll show you a drawing that kind of captures that. Essentially, what this means is that the PI3K variant is causing signal misinterpretation. So it's a bit like having a blurred vision. So to show you what I mean, let's look at these glasses here. So if you have the normal PI3K, you can see signal 1, you can see signal 2, you can see signal 3, and you can react to them appropriately, okay? If you now have the abnormal PI3K, you can't see. You need a correction glass to actually be able to resolve the signal. So you're getting the wrong information, you're not able to listen in on the different signals and do the right thing. It's not that you can't detect those signals, but you just can't tell who is who. So it's a bit like having a radio changing the frequency, but you keep getting BBC Radio 1, no matter which frequency you choose, kind of thing. Okay, so this has been a lot of work, even though it looks just like 3 simple plots. But we can now do really cool things with this technology, because we can start to do this type of eavesdropping on the PI3K communication system in different cell types. So this is work that Rob and I are planning on doing. We obviously want to do this for many more signals than just 3 signals, so that was just an example, but there are many other signals in the body. It's also important to look at the same signal but at different doses, because cells are supposed to be able to resolve different doses of, say, insulin, which is important for sugar metabolism, and different doses can result in different cellular outputs, and that's important, part of this whole communication code and system that I was talking about. We also want to look at this across different mutations. This was just an example with PI3K H1047R cells. We're also now engineering the E545K variant, for those of you who know of that variant. We also have several patient-derived cell lines that were established already back in Rob's lab, so we can study those, and we actually have them as stem cells. So stem cells are these cells that can be induced to turn into any other cell type in the body. And that then allows us to look at all of this in the specific context that is relevant for the different disease manifestations of PROS, right? So you can have muscle cells, bone cells, cartilage cells, fat cells, etc. Because although the components may be the same, the way they're used by different cells may differ. And therefore, if you give alpelisib to a muscle cell versus a liver cell, well, they have different functions, 2 cell types. So you need to really, what I want to do is understand how it then acts on these different cell types. And if we know that a patient, for instance, has a PI3K mutation in the muscle cell but no other cells, then maybe the treatment can be tuned to be more appropriate for that particular cell type. Makes sense. How is that wishful thinking? So how would we do that? Well, It's no secret that we've got powerful computational technology these days. Everybody has come across ChatGPT, I think, and other AI, artificial intelligence-driven technologies. It's not that the technology is not the problem, but it's collecting the data and then putting it into what I call predictive computational models. I should be honest, this is not something that I can just do, because I wasn't trained a mathematician or a physicist or an engineer, but I'm really lucky to be working with two really amazing senior researchers who can do this stuff and are hungry for the type of data that I can now generate. So this quantitative data where we can now start to learn what do all these different patterns mean, input, output, and then we can start to predict what if we tweak a particular node, mTOR, AKT, what happens to the patterns, what happens to the output. So really making it much more personalized and predictive in terms of understanding what is going on. So Professor Andrei Levchenko is at Yale University in the US, and Professor Krasimira Tanasova is a professor of mathematics for healthcare at the University of Exeter. And it's no coincidence that she's actually based at the University of Exeter, because that's also where the UK, the Met Office supercomputer is located. So that is a computer that is constantly crunching through tons of data, constantly live data, and making predictions so that we can get the weather forecast that Carol something, I can't remember her name, but the BBC lady who's presenting the weather forecast gives us. And you know, that relies on a lot of data, it relies on a lot of collaboration across different people, different countries. And the key thing about the weather forecast, if you think about it, is that it gives you probabilities. It cannot give you certainty, right? Well, sometimes it can tell you 100% rain. You can just look outside and predict that it's going to be 100% rain. But generally speaking, it's not always right. But it has been getting better and better and better over the last 100 years. And it's important that it's been getting better. You will be told when there is a storm, so you can evacuate, it can save lives. So what my vision really is for the next, I should say this is not going to happen tomorrow, but for the next 30 years until I retire, is really to try and build something similar in the context of PI3 kinase signaling. And what I mean by that is I want to have maps, I want to have probability maps. I want to know with with some degree of certainty whether a cell with a PI3K mutation is more likely to divide or to move or to take up more sugar. And then I want to know, based on computational predictions, what is the most relevant node that I need to tweak to make that disease cell either die or do something that is now benign. So move it away from the danger zone. But importantly, that needs to happen at the same time as understanding what happens to the healthy cells, because you're full of healthy cells that need to function and have a PI3K, functional PI3K. So whenever we want to test it, you can do this, what's called computational treatment, so in silico treatments, we'll test a drug using a lot of trials on a computer, and then you can predict, well, if I apply treatment 1, will the cell move, will a healthy cell move from a healthy zone to a disease zone? So you want to avoid those types of treatments. So it's more to stratify treatments before we even start to apply them. This is the vision, so it's very important to say this is not something that will happen tomorrow, but it will rely on all of these sort of detailed studies that we're planning to do in the lab over the next, well, decades, I suppose. But I think it's really important, because if we can do it for the weather system, then we can also do it for a biological system, but it will require a concerted effort and lots of collaborations. So that's where we are at, and Mandy, you mentioned that I've been writing newsletters. Maybe I'll offload that to Olivia very soon, because she's already shown an interest in writing pieces about prose for the public and raising awareness. I think Mandy has shared this online previously, and hopefully she will stay around in the lab even beyond this year when she's with me. But it's just to say that there is a newcomer who may join us one day, hopefully. So thank you very much for listening. I'm happy to take any questions, or also just take them during the break.
Thank you.